Hi, welcome to AP Biology. My name is Gail Pfizer. I'm from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. A lesson overview for today. Today we're going to be going over topics 3.1, 3.2, and 3.3. These have to do with enzymes, and they're located in Unit 3, which is cellular energetics. If you haven't done so already, grab your paper, pen, calculator, graph paper if you have it, and by the end of this lesson, you'll be able to explain these learning objectives. We're going to describe the properties of enzymes, explain how enzymes affect the rate of biological reactions, explain how changes to the structure of an enzyme may affect its function, and explain how the cellular environment affects enzyme activity. But first, let's have time for our warm-up. Okay, so our warm-up today is actually our homework from the last lesson with osmosis with Ms. Kate. And we have a table with some sucrose molar concentrations and some percent change in mass. Some are gaining mass, some are losing mass. And then we have this nice graph right here, percent change in mass, and then sucrose molarity on the X. So what do we need to do? We need to determine the molar concentration of the cell, and then we need to calculate the water potential. Okay, so in my classroom, we go through this 2-day event of this osmosis lab with potatoes, and they have these beautiful graphs that look just like this for me. And then my question, I come to their lab station and I say, what is the molarity? What is the solute concentration of the potato? And they go, I don't know. And not everybody, but some. Anyway, so we talk about everything from this point up is gaining mass, and everything from this point is losing mass. So what's happening right here? And then they tell me nothing. And so we discovered that if it's at equilibrium right there at that point, that is the molarity of the solution and the potato. Okay, so now that we've established that, I kind of led you to the first point of this FRQ, this very short FRQ. So now I need you to take that information, and I'm going to give you about 30 seconds to pull it together with finding the water potential of this solution, or this potato and the solution. Okay, so let's go ahead, and we're just going to, let me get rid of some of this. So we're going to insert some numbers into this equation. So we have, there is our looks like a Titan, King Titan, what's her name? Ariel's dad. Looks like his pitchfork, but his, it's our psi. And so we're going to put -1. And so why are we putting -1? Because we're dealing with sucrose. If we had salt, we'd have 2 ions involved, and that would be -2. So we're going to have -1, and then we're going to have our concentration, and then we're going to have our pressure constant. I'm running out of room. And then our temperature. So what's our temperature? If this is a Miss Kate's classroom, her classroom must be very cold, because mine runs about 22, and everybody's always freezing. So how are we going to get this? We're going to go 273 plus 18, which was 291. Okay, so now do the math, and let's see what we come up with. And I have, and let's see if you do, I have a -9.67. Okay, so let's check against the scoring rubric. All right, so yes, here we go. And we have bars. So if we did this all out nicely and it worked it out, we would have moles that would eliminate, then liters, and then we would have the K, and we would be left with bars as our unit. All right. Okay, I hope everyone scored well on that. All right, so this is what you need to know. This is a very easy, this is gonna be an easy day. And so we're just gonna talk about enzymes, and we're gonna do a couple of short FRQs and then a couple of multiple choice. So let's just start right here. The structure of enzymes includes the active site that specifically interacts with substrate molecules. So here we have, this is our enzyme, and now we have, that is our active site. And if you'll notice, it is shaped just like this substrate. And our substrate can be something that's one molecule that we're going to use through dehydration synthesis, form, break it down into two monomers or something like that. Or we can have the two monomers and we're going to then make a polymer, that type of situation. But here's our enzyme, and there's where our substrates will still fit right into that active site. All right, for enzyme mediated chemical reaction to occur, the shape and the charge of the substrate must be compatible with the active site of the enzyme. So not only does this need to be the right shape to fit, it also, the charge of this substrate and the charge of this enzyme must be compatible, okay? All right, so now let's add activation energy into this whole thing. So we have, here's our substrate, here's our enzyme, and together they're going to make our enzyme substrate complex. So what is this? What is the purpose of this enzyme? It is to speed up this reaction. It's to be a catalyst of this reaction. So how does it do that? It does it by lowering the activation energy. So that's what this E(a) is. So enzyme lowers the activation energy, speeds up the reaction, and now we have our, this would be our reactant, and this would be our product, so that particular reaction. Now, here would be the reactants, and this is the product, and we're going, like, we're breaking something apart, and now we're putting something together, okay? So same thing, we're gonna lower the activation energy and speed up that reaction. All right, so change to the molecular structure of a component in an enzymatic system may result in a change of the function or efficiency of the system. So I'm not really addressing denaturation yet, okay? So if with this active site is supposed to be this shape for this substrate to fit, if this was to have some conformational change, this substrate is no longer going to fit into this enzyme. Okay, so it's not going to function as it was originally designed to do. All right, so now let's talk about, I want to focus on the activation energy that's in this graph. So if we were to, so this is the energy that we need to invest into this reaction. So this line right here, let's get a brighter color. All right, so this line right here would be the energy that we needed. So we're just coming to this point for if we had an enzyme. If we didn't have an enzyme, then we would need this much energy invested before we could get to the product. So here is the enzyme lowering the activation energy. All right, so here we are, denaturation. That seems to be something that kids remember from early biology. And denaturation of an enzyme occurs when the protein structure is disrupted, eliminating the ability to catalyze reactions. So here we have our enzyme, and this is our active site, okay? It's a very elaborate active site. So our enzyme goes in there. Now, if we were to denature this enzyme, and we'll talk about ways on the next slide, if we were to denature this enzyme, this shape is then no longer there for this enzyme to work on this substrate. All right, so environmental temperatures and pH outside the optimal range for a given enzyme will cause changes to its structure, altering the efficiency with which it catalyzes reactions. So again, here's our enzyme, here's our substrate. So there's our substrate. And so now we have our enzyme substrate complex. Now, if we were to apply too much heat, or if we were to get, there's an optimal pH that enzymes work in. If we were to go too basic or too acidic, then we could denature this enzyme. Okay, so pH and too high of temperatures. All right, so speaking of the pH, let's talk about the hydrogen ion concentration that is in pH. Let's start this way. All right, so let's look at our protein structures. Remember, we had 4 structures. We had a primary, and then in our secondary, I think I had black dots on the last time we did this. All right, so here's our hydrogen. Our hydrogen bonds are between these amino acids, and it's giving it this form in this alpha helical shape. And then we also had them in our beta pleated sheet. Both of these are secondary. Hydrogen bonds make them have this particular shape. And there were hydrogen bonds that held our tertiary together. And if we disrupted the hydrogen ion concentration and made it too acidic, made it too basic, that's going to disrupt these hydrogen bonds that are holding it in this particular shape. So this would no longer be a functional enzyme if we disrupted the shape with pH. And speaking of pH, I know this looks crazy, but so I scribbled out this craziness on my board one day, and it was about how to understand what are you talking about when you're talking about pH. So when I did this with just, in pen on the whiteboard one day, and then there was some lights went off in some people, or some lights came on in some people, and they like, oh my gosh, I get it. So let's talk about some facts about pH. pH is based on 1 mole of hydrogen ions, and the pH measures how many hydrogen ions are in a solution. And pH is the negative logarithm of a hydrogen ion concentration. And these brackets right here, okay, that the concentration is in there. Now, each 1 unit change in a pH scale is equal to a 10-fold change in hydrogen ion concentration. So let's take, for instance, a pH of 5. A pH of 5 is 10 times more acidic than a pH of 6. But a pH of 5 is 10 times less acidic than a pH of 4. I hope I said that right. All right, so what does this mean? With a pH of 1, there is 1.1 moles of hydrogen ions in the solution in 1 liter. And so this means that there's 1/10 of a mole, and then we have this 10 to the negative 1. In a pH of 2, we had 0.01 moles. So that was 1/100th of a mole of hydrogen ions is in a pH of 2 solution, and there's our -2. Okay, so I'm not going to go through all these, but you get the point of how this all of a sudden helped some students understand what this means. I hope it helps you. I had this a little bit different, and I have to give a shout out to our AP Calculus teacher that keeps me in line and tells me, no, I just can't make up my own math. And so she made sure that everything was in the right order here. Thank you, Ms. Tanya Hickson. All right. So the relative concentrations of substrates and products determine how efficiently an enzymatic reaction proceeds. Increases in concentrations of substrates and enzymes, so increases of substrates and enzymes can increase the rate of the enzymatic reaction. So we can increase the rate with these 2 things. All right, so however, as level of product increases, feedback inhibition can result. So if we're using an enzyme and we're making a product, then if we get enough product, there's no reason to continue to make that product. So we need to shut that enzyme down. And that is a negative feedback mechanism to maintain homeostasis that could happen. All right, so higher environmental temperatures increase the speed of movement of molecules in a solution, increasing the frequency of collisions between enzymes and substrates, and therefore increasing the rate of reaction. So this is my attempt to show that at low temperature, we have low collisions because they're not moving as fast as they would be, the molecules aren't, as if it was at a higher temperature. So more collisions can happen, so the rate of reactions can then increase. All right, so competitive inhibitor molecules. This is one of my favorite topics. Competitive Competitive inhibitor molecules can bind reversibly or irreversibly to the active site of the enzyme. So here is our active site of our enzyme that fits our substrate perfectly. Now, our substrate cannot get to the active site now because now there is another molecule with the same shape that is blocking this substrate. That's called competitive inhibition. It's stopping it, it's competing for the same spot. All right, non-competitive inhibitors can bind allosteric sites, changing the activity of the enzyme. So here we have, again, our substrate, our enzyme, but now we have an allosteric site. So if we have a molecule bind this allosteric site, if you notice, look at the active site normally, okay? When this binds, it changes the shape of our active site. And now our substrate can no longer fit into our active site because the allosteric substrate is in the allosteric site. All right, this is some work from my classroom. These are models of competitive and non-competitive inhibition. And so if you'll notice, and this little guy, here's his active site, and there's his substrate right here. And then she also, there's a allosteric site. And so when actually they work, because that's part of the requirement, they have to work. So when she puts this allosteric, the substrate for the allosteric site in, it actually clamps down on this little guy's mouth, and then the substrate can't fit into the active site anymore. And then we also do this toothpick lab that a lot of people do in their classrooms. And so at one lab station, there'll be one person with this huge pile of toothpicks. Everybody, all groups have the same amount of toothpicks, but the group members are different in each group. So there'll be, in your hands, the individual is the enzyme, and so your hands are the active sites. So one person and 750 toothpicks to break will be much slower than 6 people with breaking apart 750 toothpicks. So we take the count every 30 seconds or so, and then we come up that the rate with more enzyme is much faster than with the less enzyme. Another lab that we do, this is investigation 13, and this has to do with ternate peroxidase. And what we do is students use ternate peroxidase to break down the hydrogen peroxide. So what happens is the oxygen, so when H2O breaks down, so when H2O2 breaks down, you're going to have an oxygen gas. It's going to be a product of that. So in the presence of oxygen, this indicator, guaiacol, which smells horrible, actually will bind to the oxygen, and when it does, it turns brown. So when it's brown, then we can tell there's oxygen there. And so the more brown, the more oxygen, which means the more peroxidase, that the more H2O2 that is being broken down. And then after we establish our baseline of what it looks like under normal conditions, then we start changing the pH. And this is a student's graph. You can see that nothing's happening at their pH of 3 and their pH of 10, but it looks like 5 took off and probably ended up to be the fastest rate of reaction that happened, is at a pH of 5. So that was optimal for ternate peroxidase. All right, and then I know this is a whole week of enzyme labs. We have so much fun with enzymes. So these are yeast balls, and we make them with sodium alginate. And so we put them in this little container. This is like a water bottle that's that little tiny half water bottle size. Anyway, so we put the yeast balls and some hydrogen peroxide as the substrate in there. And there's this little half pipette right here, and then there's this graduated cylinder. And what's going to happen is this yeast, the enzyme within the yeast is going to break down the hydrogen peroxide, and then oxygen gas bubbles will be formed, and they go up, and then there's this displacement of the water in the top of the graduated cylinder. And then measurements are taken every 30 seconds or every minute, and then we see how fast this is at working in this condition. You can make the water colder, warmer, change up the amount of substrate, change up the amount of yeast balls that you have in the apparatus is what I call it. Okay. All right. So that's all for content and for science. We're going to work on some science skills. So a lot of this is going to have to do with leading to, actually, we're going to be leading to experimental design. So 3CB, identify experimental procedures that are aligned to the question. And task verbs that we're going to use today, we're going to see a lot of identify and determine and discuss. So first, let's do one together. And so we're going to identify. And how many? We're going to identify 2, and we're going to identify 2 what? 2 environmental factors that can change the rate of an enzyme-mediated reaction. And then we're going to discuss how that would affect them, as in, is it going to make the rate increase, or is it going to make the rate decrease? So I'll give you about 20 seconds to jot down some words really quickly to see what you think. All right, you have a couple? All right, let's look. So for identification, you could have said temperature, pH. You could have also said salinity, inhibitors. Stirring and mixing would have never came to mind personally. Pressure, you can change the pressure in your classroom just by opening the door to the hallway. Oxygen and light. In discussion part of this, you should have said there's something missing on this scoring guideline. If this temperature increases, then this rate should increase. That little arrow is missing there. Okay, so temperature increases, rate increases. That's one discussion point. Temperature decreases, rate decreases. High temperature causes denaturation. Other factors that you could talk about, improper folding, change of the active site, some altered kinetics that have to do with the temperature altering the rate of collisions. You could have also talked about the pH and salinity and active site changes related to all of these, okay? All right, so now let's graph. We're going to do 2 things. We're going to graph, and then we're going to determine an optimal temperature for respiration of the yeast. So here we have a table. Here's our temperatures in Celsius, 0 through 70, and our number of bubbles of gas produced per minute. And let's see what it says up here. Yeast cells are placed in an apparatus with a solution of sugar, a major nutrient for yeast metabolism. The apparatus detects bubbles of gas released by yeast cells. The rate of respiration varies with the surrounding temperatures as indicated by the data below. So I'm going to take this to the next page, and I need you to grab whatever you're graphing on, and we're going to kind of have a graphing, parallel graphing together moment. I don't want you to copy me, look at me. I just want you to on yours, and then we'll compare both in the end. Okay, I have all of this data on the next page as well. All right, so I'm going to get going. First, okay. All right, so. It is pouring rain at my house right now. Okay, I'm finished. I could leave it this way. I can do it a couple of different ways also. If you could use data points, you can use a line, or you can use bars on this particular graph. Now, since we're dealing with enzymes, and you often think about enzymatic rates, then I might have drawn a line for this graph, but I know I would have probably just done data points. So let's look at the scoring guidelines. See, that's exactly what it's supposed to look like. So now we don't need a title, but let me say this. You must label your y-axis completely, okay? It must say what it's supposed to say. And if you titled a graph, then if you left something out over here and it was up here, then that would be okay. And then we have our x-axis, we have it properly labeled. We had our scaling. Did you notice me scaling? I knew the graph because I practiced earlier to make sure I could do this with you. And so I already knew that I couldn't skip lines or anything like that. All right, so correctly plotted points. Points may or may not be connected with a line, and a bar graph would have been acceptable. And our optimal temperature was at 30. Now I know that you're not going to be doing a graph on your test this year, but you're going to probably be analyzing one. So if you know how one was built within a topic, then that's going to help you be able to analyze it in that same topic. And then possibly, if we're in a few minutes, we're going to talk about rate, and then we'll talk about possibly rate on the exam. Okay, so let's look at these multiple choice. We have a hydrogen peroxide solution. We have 1%, 5, 10, 15. It looks like we're increasing our hydrogen peroxide solution. Average time in seconds for disc to float, 30 seconds, 25, 20, and 10. So looks to me like the higher solution, the faster floating. So let's read. To study the actions of the enzyme catalase on hydrogen peroxide, students performed the following experiment. Catalase was extracted from potatoes by blending raw potatoes in a blender with cold distilled water. The filtrate was stored on ice. The following hydrogen peroxide solutions were made: 1%, 5%, 10%, and 15%. Filter paper discs were soaked in the catalase filtrate and dropped into the beakers containing the various solutions. The activity of the enzyme was measured by the amount of time it took for the disc to float to the surface of the solution of the bubbles produced by the reaction. The following data were obtained. So let's talk about what happened. So here's our beaker, and we have some disc soaked in hydrogen peroxide. And when that hydrogen peroxide is then be broken down by that catalase, then there's going to be oxygen gas bubbles that are going to be underneath these discs, and it's going to lead these discs up to the top of the beaker in the catalase. Right? Okay, that's what is going to happen. So let's read the question. If the potato was boiled for 10 minutes and cooked for 10 minutes before, and cooled for 10 minutes before tested, the average time for the disc to float to the surface of the hydrogen peroxide solution would be, so it's hydrogen peroxide solution, they're soaked in catalase when they go back down, if I got that backwards just a second ago. All right, so less than 1 second, 5 seconds, 10 seconds, 30 seconds, or more than 30 seconds. Okay, this was a clue. They were boiled. So what do you think it's going to take? I hope you said E. It's going to take more than 30 seconds because we denatured those enzymes. All right, next question. We have a graph. We have a, excuse me. We have 3 lines. We have a pepsin line, a chymotrypsin, and a trypsin. And so pepsin is in solid, chymotrypsin is in dashed, and bold is the trypsin line. Okay. Protein digestion in humans is primarily carried out by 3 enzymes. Pepsin is found in the stomach, which is pH of 2, where it aids in the breakdown of large proteins into smaller peptides. Where trypsin and chymotrypsin are found in the small intestine at pH of 8. excuse me, where they aid in further breakdown of the proteins into amino acids and dipeptides that can be absorbed into the bloodstream. Graph 1 shows the effect of pH on the activity level of the 3 enzymes. All right, so what's our question? Which of the following best predicts how the structure and function of pepsin will change as it enters the small intestine? So we're going to go from a 2 to an 8. All right, so pepsin will not change in shape? No. Pepsin will not change. I don't even need to read the rest of that. Pepsin will not change in shape again. Pepsin will change in shape because of basic environment of the small intestine. Therefore, its enzymatic activity will decrease. Okay. Pepsin will change in shape because of the presence of trypsin and chymotrypsin in the small intestine, both of which act as competitive inhibitors. Are they acting as competitive inhibitors? No. So our answer is C. All right. I'm gonna stay out of this one as much as possible. All right, the enzyme peroxidase is found in many organisms. It catalyzes the breakdown of hydrogen peroxide into water and oxygen gas. The rate of peroxidase activity at different pH values was assessed by students in the lab. The students' results are shown in the graph. So here we have relative peroxidase activity. We have pHs of 3, 5, 7, 9. And the question's asking you, if the experiment is repeated at pH of 11, let's put 11, the observed activity level of the enzyme will most likely be A, the same as the level at pH 7, the lower than the level at pH 9, greater than the level at pH 9, or between the levels of 5 and 7. All right, I'm not going to answer that one. I'm going to let you answer that one. And I'll give you 5 more seconds to think about it. Okay, and I hope you said what? B, lower than the level at pH 9. Okay. All right, so here we're going to have some guided practice. We're going to graph again, and we're going to, because that rate is what this is all about, is in enzymes. All right, so we're going to graph the data, and then we're going to calculate the rate. This is something that could easily be a graph that's on your exam. I know nothing about your exam, but I do think about what's something easy that an easy calculation could fit into, and rate is one of those. So this is worth your time. An experiment was conducted to measure the reaction rate of the human salivary enzyme alpha amylase. 10 milliliters of a concentrated starch solution and 1 milliliter of alpha amylase solution were placed in a test tube. The test tube was inverted several times to mix the solution and then incubated at 25 degrees Celsius. The amount of the product maltose present was measured every 10 minutes for an hour. The result was given in the table below. So now we're going to graph something, but we're dealing, it's right there. We're dealing with time. So I think you know what kind of graph to make with this, and it's going to be an enzymatic rate. So we're going to graph, and then we're going to calculate the rate. We'll look at the rate period between 0 and 30 minutes. So let's go graph this. We can graph it together. Here is our time, and there's our micromolar concentrations of our maltose. Okay. Is that all I needed? Yes. And now I need just past 12. Okay, and 0 at 0. 5.1 at 10. 8.6. And 30 is 10.4. And 11.1. 11.2. And 11.5. All right. Okay, does your graph look something like that? All right, so now let's do the rate. It's set between 0 and 30 minutes, right? So if we go 10.4 micromoles over 30 minutes, how much do we have per minute? You go ahead and do that calculation. And we have 0.35 micromoles per minute. Now, this would be an easy skill to be tested on. You could still do a calculation. You could still be involved in a graph. So I'm going to make sure my students are up on their rates and what's going on with enzyme lines. I think is very important. All right, then this is what it looked like. Our graph did look like that. We had correct orientation of independent was the time, dependent was the maltose, and we have a units and intervals. We scaled it. I'm an obsessive scaler, and correct graphing of all data points, and then our correct setup. And there we have, our calculation fell in the range that was acceptable for the rate calculation. All right, so explain why a change in the reaction rate was observed after 30 minutes. I'll give you about 15 seconds to think about that and jot something down. Okay. So, did you say that the reaction rate slows and levels off? And then why? So because it said explain, so that was how or why. So the rate slows as the substrate concentration declines. If you're using up all the substrate, then there's nothing else to break down or put together. Now, the enzyme inactive by about 40 minutes. If your enzyme lost its activity over time, became labile enzyme, or if we had product inhibition come into play with this, that can also be an explanation of why it slowed down. All right, debrief and summary. Today's takeaways. Enzymes decrease activation energy, increases in substrate. Enzymes and temperature can increase the rate of reaction, but that temperature and pH, if it's the wrong temperature, if it increases too much and the pH is not in the optimal range, then denaturation can occur. You should also be able to describe the properties of enzymes, explain how enzymes affect the rate of biological reactions, explain how changes to the structure of an enzyme may affect its function, explain how the cellular environment affects enzymatic activity. And I always want you to be able to describe it, explain it, and tell me what you would do without it. But now I want you to tell me what would happen if you changed it. So some common errors and misunderstanding, and this will become more clear when we talk about when we get into photosynthesis and cellular respiration. One enzyme is not responsible for the end product in a metabolic pathway. The product of one reaction is going to become the reactant of the next reaction. And then I know that sometimes allosteric inhibition it's always interference, but it's not always a negative thing that's happening. Negative feedback has to happen. It's a necessary part of homeostasis, where our bodies can't continue to make continue to make a product that we don't need and waste that kind of energy. And then let's see what else there was. Oh, I know what else I wanted to add. It was about temperature. I forgot to. So cold does not denature, okay? That's why we put it in a refrigerator, to slow the enzymes down so they're not working before we need them to work in lab. It's also why you put things in a refrigerator. One of the reasons. Okay, so your homework tonight, there's two graphs involved in this. The effects of pH and temperature were studied for an enzyme catalyzed reaction. The following results were obtained. So we have enzyme activity and temperature. It looks like we have an optimal temperature, a crooked line, and enzyme activity and an optimal pH. So what you're going to do is you're going to describe a controlled experiment that could have produced the data shown for either temperature or pH. And be sure to state the hypothesis that was tested here, but I want to change this up a little bit. There's not going to be one right, exactly the same answer. Okay, so this is what I just want you to have some experience or some practice with some experimental design. This could be something else that could happen. A graph was given to you, and then you're all of a sudden having to putting all the pieces together. That would make a great FRQ. All right, so hints, what are you measuring? Make sure you cover this. What are you measuring and how? You will need an independent variable, a dependent variable. You'll need a control. Go ahead and put together a null, okay? And then also your alternative. So have all those pieces together and of what a good experimental design to get to this would look like, okay? And that will be, oh, I'll talk about that in a second. All right, device and internet access. Do this by April 24th. We know this is from the College Board. We know that not all students have access to the internet or a device. We're working on solutions to help students get what they need to show their best work. If you need mobile tools or connectivity or know someone who does, you can reach us directly to let us know at cb.org/tech. All right, one more slide. And thanks for watching, and I hope you'll join us again. We'll be sure to complete your homework question for tomorrow, and then Ms. Kate will go over that first, and her lesson tomorrow or after this lesson, if you're not watching them in a day-by-day regimen, then she's going to talk about photosynthesis. So have a good day and come back and see us. Bye.